Welcome to Decoding the Data Ecosystem, a podcast with valuable insights, inspiring stories, and practical advice to guide you on your journey through the world of omics research and how the NIH Common Fund data ecosystem can support that journey. I'm your host, Dr. Allissa Dillman, and I'm the Training and Engagement Director of the CFDE Training Center. My background's in bioinformatics, and I've been working in bioinformatics and data science training and engagement space for almost the past 15 years. I'm really passionate about lowering the barriers of entry for these fields through creative engagement activities such as these podcasts. Now let's get to the show.
Today I'm here with a couple of folks, and so we're going to talk a little bit about glycoscience. We have Raja Mazumder, who is a professor in the Department of Biochemistry and Molecular Medicines in the School of Medicine and Health Sciences at Georgetown University. We also have René Ranzinger, and he is an assistant research scientist at the Complex Carbohydrate Research Center. And we also have Michael Tiemeyer, a joint director of the Complex Carbohydrate Research Center and Center for Molecular Medicine. He's also a distinguished research professor at the Department of Biochemistry and Molecular Biology at the University of Georgia. So hello, everybody. Really excited to have you on. And I would love to start us out with telling us a little bit about your background and how you became interested in glycoscience.
Yeah, I'll start. This is Michael Tiemeyer. I was trained actually in a neuroscience graduate program. I was very interested in how the brain's put together, how it develops, and how it works. But when I was an undergraduate, I sat through a mandatory biochemistry lecture by a professor named Saul Roseman, who was a giant in early glycobiology, carbohydrate chemistry. And I kind of fell under the charismatic spell of what glycoscience was all about. He was talking about how glycans on cell surfaces allow cells to interact with each other, all the way from sperm interacting with eggs and all kinds of other sorts of things. And it just grabbed my attention, and trying to think about how to apply that to the brain kind of got me launched in my career.
My name is Raja Mazumder, and I have always been interested in how biomolecules function and how these functions evolve over time. So early on in my career, most of my work focused on genes and proteins. Genomes were getting sequenced at a rapid pace, and that was a really nice way to see how in different organisms these genes and proteins evolved over time. But as I studied that and I studied those systems, I realized that the post-translational modification play a huge role, right, in regulating protein function, diseases, and so on. And slowly got pulled into glycosylation because it's one of the most important biologically important functions, modifications. And it was natural for me to start exploring how glycans influence protein behavior or diseases and so on. I'm an informatician. So the challenges around it, how do you integrate that data? How do you connect it to different data types?
Hello, my name is René Ranzinger, and I'm actually a computer science by training. And I didn't really have to do much with biology and chemistry at all during my study. But when I finished my master degree, I started working with Dr. van der Liet at the German Cancer Research Center on a program for 3D modeling of molecules. So that got me a little bit interested in that bioinformatics aspect. And then I started my PhD with Dr. van der Liet and focused on glycoinformatics tools and database development. And I found that actually very interesting because compared to other fields in bioinformatics, glycoinformatics was really in its infancy. There wasn't many databases around, many tools around. So it was really interesting to develop new tools and apply even established algorithms for these new fields. And I have been working on databases and tools ever since, and also worked a lot in standardization of information. And that's also one of the things that we have done in GlyGen.
I love that you all came at glycoscience from very different angles, right? You kind of came from different backgrounds and different fields of study, but you all sort of ended up in glycoscience. That's really cool. I'd love to have you give us a little bit of an overview of the GlyGen program and its main goals for our listeners.
Okay, I think I will take that question. So if you're looking around in bioinformatics databases, and glycans are related to proteins and genes, so there are tons of databases out there, and every database has certain focus and parts of the information, so pieces of the puzzle. So we have proteomics databases like UniProt, which tell you a lot about the proteins and a little bit about the PTMs, but doesn't talk at all about glycans. And then we have Glycoscience databases, which have all the information about the glycans and so on, but very little about the proteins. And then we have genomics databases. So, and around 2017, when the GlyGen project started, that was the starting point. And the motivation for the GlyGen project was, okay, can we take all this information which is related to glycans and glycoscience and bring it together in one place? Harmonize the information, standardize the information, to integrate this information and do some validation on that data. And then once we have that consistent representation of all this information, can we make it accessible to the community? And that's really what GlyGen is. It's this integration of all this data that we can access into one resource, making it accessible, but then also always providing links back to where did that original data come from? Like, what are the papers talking about a certain annotation, or which other resources, databases provide that information? And also, since we are not completely integrating all data from all these databases, give a way of linking back to the original databases, which have more information that people might be interested in. So that's really what GlyGen is. And as I already said, the main goal is really make the data accessible and provide a more holistic view on glycans and glycoinformatics in the context of proteins and genes.
So I'm definitely more of an RNA biologist by training. So the world of glycoscience is definitely new for me. And I think it might help to kind of frame things by asking, like, what kinds of scientific questions become easier once GlyGen makes glyco knowledge more findable and computable?
So, Alyssa, I'm going to say something that us glycoscientists always like to say to molecular biologists and RNA people, and that is that you actually are a glycoscientist too. RNA is just a polymer of ribose, which is a carbohydrate that's decorated with these nucleotide bases. But all of molecular biology really revolves around carbohydrates in one way or another. But I guess to answer your question, I would kind of take a step back and talk about some of the ways that knowledge has grown in the biological sphere. And some of those approaches are sort of classic genetic screens, and even some of the newer CRISPR-Cas genome-wide screens that identify genes that are likely important for the biological functions that have been screened for. Many times in these screens, people end up with molecules that are predicted to be at the cell surface, or transmembrane proteins, or they're secreted proteins, and now they've been tied to some sort of important regulatory activity, regulating cell growth or migration, cellular differentiation, structure of a tissue. And so geneticists are left with these wonderful glycoproteins. And before a resource like GlyGen, these folks might be thrilled that they've got this new function, but don't know what to think about this molecule they have in their hands, not really understanding how it's modified with glycans extensively sometimes, and what those modifications might mean. So now these folks can ask questions about, first of all, is this cool new protein they've identified, is it modified with glycans? Is it glycosylated? And what type of glycans are there? What sorts of sort of structural and functional glycan motifs are found on that protein and how might they regulate the function of the protein. So GlyGen allows people that are glyco-naive to begin to understand the impacts of glycans on their particular molecules of interest. I'm the director. The CCRC is a collection of 16 investigators and 300 scientists and trainees. And the investigators here, we always tell the story of how we're sitting in our office and it used to be phone call, now it's usually an email that says, I've got this thing that's a glycoprotein. I run it on a gel and it's a big smear. What do I do next? Well, now we can help people understand some of what they can think about doing next by understanding what's on their protein. So I think that's some of the new sorts of questions that GlyGen makes more accessible.
Well, I definitely love hearing that the field of glycoscience is so opening and welcoming of a community. That's nice to hear, and I will definitely feel like I belong more now, given your explanation. So, kind of following up with that, is there like one misconception about glycans and glycosylation that you hope that GlyGen can help correct?
Oh yeah, I'll take this one. Yeah, so when we talk, when glycoscientists go out and give talks, sometimes we do a real disservice to ourselves because we'll talk about how complicated these glycan structures are. And to us, that means they're beautiful. But we start by saying these things are so complicated. And that attitude again stems from sort of the inherent chemistry of the molecules and how they're put together. But the disservice is that you don't really need to be a synthetic organic chemist or a glycobiochemist or a structural biologist really to kind of understand the beauty of the biological roles of these molecules and the diversity of the possibilities that glycosylation has for changing cellular and biochemical activities. So this misconception about how complex these molecules are or how complicated these molecules are, I think scares people away. And so GlyGen tries to present what are sort of the broadly accepted ways to sort of simplify representations of glycans. These are things that have been developed by our community over the years. And being able to reference those and understand those, I think, can help us kind of demystify our domain and help us engage with other people that have thought that this is too complicated. But it's really not. It's just really beautiful.
That makes me think a lot about just like structures of, molecules in general, right? Things like even DNA and RNA have these complex structures. And I feel like as biologists, especially like molecular biologists, we kind of get focused on one aspect or the other, and we forget that like we need both of those things to kind of understand function. So that's a really good point that you make. I do have a question around kind of the glycan structures in general. So I know that glycan structures can be ambiguous or only kind of partially known. So how does GlyGen represent uncertainty without sort of overclaiming that like this is the absolute known or absolute answer for this structure?
Yes, so I can say something about that. And that's actually a very interesting problem and also something which might be different from other fields, that when we are talking about the glycan, very often we don't know the exact structure because of, the amount of material that you have or the experimental technique that you use doesn't provide you with all that details. So just to give an idea, very often people publish compositions. So something like my glycan consists of 4 hexoses and 3 hexnugs. So if you think about proteins or genes, nobody would publish like, oh, I found this protein with 120 asparagines and 70 serines or something like this. You would always have a sequence. So for glycans, that's a little bit different. And that might be also something like Mike said earlier, giving an additional complexity to it. But from our point of view, any kind of representation, any knowledge that you have about a glycan, may it be a composition or maybe you just know how the monosaccharides are linked together without knowing the exact linkage positions or something like that, any kind of level of information that you have is valuable information that we need to capture. And in GlyGen, we really capture exactly this level of information. So if you have missing information, that's okay. Our sequence formats, our representations of the glycans can handle that, and you can store that information. As I said earlier, we are integrating information from all these other databases. And one thing that we do is we keep exactly that level of detail that the original publication or database provided and use that level of detail in GlyGen. So we wouldn't add additional information or making assumptions. We just keep that information as it is. And that has the advantage, it really reflects what, for example, if you do a curation of a paper, will really reflect what was published in that paper without reducing or increasing the level of information there. But on top of that, since we know, composition or, incomplete structure doesn't really exist in nature. There is, a defined molecule in the end. We have worked on ways of linking these structures, these different information detail or level of details together so that we can say, okay, we all belong together and we might be representing the same glycan structure, even if based on that level of detail that was published in the paper, for example, does not explicitly say that. But if you make this kind of linking together and also propagating information, for example, somebody published a glycan structure in a human species, for example, we may propagate that information to the composition of the glycan structure as well. But if we do that, it will be explicitly stated in our database where this information came from. So I hope that kind of helps understanding that we can handle this different level of details, but we are not trying to change the level of information that we get from the original databases or papers.
That kind of leads me to ask, like, how do you deal with like naming these things? I feel like you mentioned something called GNOME before. Does that help with this?
Yes, so to some extent. So glycans don't really have a name, but around 10 years back, the community agreed on a common standard how we identify glycans. So there's a glycan ID scheme, which is provided by a database called glycan. And that is really the main identifier we are using for glycans. And then that identifier is also used in GNOME to link the different structures to each other. So if you think back what I said earlier, there's a glycan and then there's the composition of that glycan, and each of them might be coming from a paper. Each of them will get a unique ID in the glycan registry. But then GNOME, for example, allows us to link them together saying, yes, we are belonging to some together.
Okay, I see. Thank you for that clarification. So what is it about glycoconjugate data that's harder to integrate than other types of omics? Is there like a hidden complexity that a lot of people miss, especially if you're kind of coming at it from, say transcriptomics or, genomics?
Actually, this question is related to what René just now mentioned in response to your previous question. So for many other post-translational modifications, the question is relatively simple, whether a modification is present or absent at a particular site on the protein. But for glycosylation, it's very different, right? So at a single site, you can have a large diversity of glycan structures attached under different conditions, different cell line, different patients or individuals and so on. So we cannot just say on this protein, such as EGFR, on site number, X, you always see this structure, right? So there's a lot of variability that we can see, the branching pattern and so on. So the complexity isn't just the modification itself, but the structural diversity of the glycans. And to really understand the biology, one needs to appreciate the different glycan motifs, the branching patterns, and the various types or subtypes of glycosylation events. And that structural diversity is what makes glycan data much harder to standardize, compare, integrate across data sets compared to other omics data.
I see. So it really has that dynamic nature. Although it does kind of make me think, because again, RNA biology, up splicing, right? Because you can have all these different odd, rearrangements and permutations that you may only see in specific tissues, specific diseases, or even in a specific person, which makes it really hard to kind of define, like, exactly which splice isoforms there are and where they are and things like that. But obviously it's slightly different, but it kind of makes me think back to the things that I know.
Yes.
Okay. So I'm curious to know kind of who, at least for now, is the primary user that you're building for. Like, who was sort of the initial, we hope that people will use this, these tools, these databases and the other resources that you've built.
So I can say a little bit about that. We are trying to be really broad in terms of the users that we want to address. So if you think about use cases, for example, you may have a bench scientist that studies a protein or found a glycan and now wants to just look up, okay, what information is available about that glycan, or, what do people know about my protein and its glycosylations? This is people that we are helping with our web portal. So we have multiple different ways how you can access the data. One is our web portal where you have, you go there with the browser, there's a search interface, you can look for information, you can look data up, you can download information. So bench scientists, biologists, chemists, also on, we can use this as an easy way to access that information. But in addition to that, we also want to help bioinformaticians or people that want to do data mining, machine learning, to work with this data. So we have APIs, Sparkle endpoint, and also a data portal where these people can, submit queries and get custom data, or just download all the data that we have and use it. We are also trying to go in the direction of helping clinicians, for example, by providing dedicated search interfaces for diseases, which is more their entry point to come from a point of view of a biomarker or a disease and then want to look up information. So we are trying to address many people. So I am not sure there is really a primary user.
That's fair. So it sounds like success looks different for different kinds of users, and you've got kind of different kinds of tools for those different kinds of users.
Yes. So success is not easy to quantify for this kind of projects. But what I would say is if people that look for information can find that information easily, then that's a success for them. And especially for bioinformaticians that, want to download the data or do some data mining, one advantage of the data sets, as Raja mentioned earlier, is this integration of different pieces of information that otherwise we would have to go to 10, 15 different databases and collect all that information. While using our APIs or our data website, we can just get that in one go. So that's also a way how I would say that's a success for GlyGen if we can do that. And then, for everybody, it saves some time. If I don't have to go to 10 databases, or if I can just, use an easy to use search interface and get the information I'm looking for. So that's another way how we can measure success.
Awesome. That sounds like you've got just like a lot of different tools and different ways to access your data in a lot of different ways. So I'm curious on if you could kind of walk us through sort of a concrete workflow that typically happens with the GlyGen resources.
Actually, I'll give you maybe 2 or 3 workflows. So if I kind of build off the vignette I mentioned earlier, where a geneticist finds a new biological function for a gene or the protein that it encodes, geneticists can ask what sorts of glycans have been reported on this protein. And more importantly, I think at higher resolution, where on the protein have glycans been reported to be, what sites? And then the geneticist can get some information related to those sites. As Raja mentioned, how diverse are the glycans that have been found on that site? That's a sort of concept we call microheterogeneity. How heterogeneous are the glycans that you find on that site? And then also what sorts of variants have been described at that site. And that can lead through GlyGen to literature that describes something about the site and the variants and phenotypes that might be associated with those variants. So there's the workflow of a geneticist or somebody coming to GlyGen with a gene or a protein and trying to get more information about that protein. But we also have entry points for glycochemists, for people that have a glycan but don't know anything about what it might be attached to. So they just may have extracted all kinds of stuff from a tissue or cell source, and they've isolated some glycans, and they've spent months and years characterizing what those structures are, and now they want to take the next step. And they can begin to ask questions at GlyGen. Given that glycan, they can ask questions about what are the enzymes that make that glycan in a tissue, in an organism that we might have data for. And also then what proteins has this glycan that you've identified been described to be connected to? And what's the function of that protein? And it begins to build a data set that allows you to generate hypotheses related to the function of the glycan or how the glycan influences the protein that it's connected to. Or someone might come to GlyGen, sort of another workflow, having identified an enzyme that's simply called a glycosyltransferase. And you might have no idea what that means. There are hundreds of glycosyltransferases in most organisms. Bacteria have tons of them as well. So just knowing you have a glycosyltransferase can be minimally helpful. But now at GlyGen, with that gene name, you can explore what that enzyme might do, what sorts of biosynthetic pathways does it interact with. Again, kind of giving you more data to set up the next set of experiments to understand your biological functions.
So from sort of both of these questions, I really get the sense that you can really kind of approach glycoscience from a lot of different angles. And it sounds like there's also a lot of just dynamic nature of glycoscience. I'm curious, how do you handle then like data quality and conflicts across sources? Right?
You're absolutely right. I mean, data quality and provenance are absolutely critical for any resource or knowledge base. And the first step about data quality, or even thinking about it, I feel, is data harmonization. So one first needs to map entities so that the same gene and the protein or glycan from different sources can be aligned correctly. So once you do that, then you can start thinking about, okay, and what kind of information I'm getting from different resources or papers and other sources. It kind of sounds straightforward, but it's quite challenging because different authors and data, at least in glycoscience, because different authors and databases often use different naming systems or namespaces for glycans. And earlier on, René mentioned about glycan, which is giving this ID to glycans, which has helped quite a bit. But then also GNOME, which is an ontology for the structures, glycan structures, allows us to understand, are the two people in two papers talking about the same thing or different things or not? Then there's this other complication, which I don't think is just for GlyGen, but for many resources, is lineage, data lineage. So sometimes one resource imports data from another, which itself imported from a third source, and so on. So tracking that chain of origin can be quite difficult. And definitely in the front end for users, they might find it really hard to figure out, okay, where is the evidence for this connection I'm seeing between this glycan and this protein and this disease, let's say. So in GlyGen, we are trying to address this by using IEEE open standards. It's a standard called BioCompute. So we capture and document that provenance. By doing that, we can track where the data came from, what transformations were applied to it, if any. So even though we do not change any data, incoming data, but we do add annotations to it, right? So for this particular thing, I'm calling them, let's say, what type of annotations we added, how did we add them, and what evidence supports each annotation. I think that's the key. That way users can see not just the information, but they can also see the supporting evidence behind it and make their judgment calls. To generate a new hypothesis, like, okay, so this evidence does not clearly say that, what I'm looking for. So maybe more experimentation is required to confirm this. So, right. So hypothesis generation is one of the key things we hope GlyGen users are using GlyGen for.
So it's been really cool to kind of hear just all of the tools, the resources, the data, the standards that you all have been working at GlyGen. And I'm really interested to kind of hear a little bit more about what's next for GlyGen, maybe a feature or capability you're really excited about, and maybe also a challenge that you really are kind of feel is quite hard to solve in this space.
Yeah, I'll describe something that I think we're rather excited about. So the gene and protein world has this ways to talk about the functions of genes and proteins in a way that machines can read that, sort of controlled terms, so that you can share knowledge about function. Go gene ontology is an example of how you can describe functions of genes and proteins in a controlled informatics rich way. The glyco world doesn't have any sort of similar ontology to describe glycan functions, but it's really important. Think about how an ontology that describes glycan functions would allow people to come into the glycoscience world, into GlyGen, with a function in mind, right? You don't have to have a protein, you don't have to have a glycan, but you have a function you want to learn about. And then we could tie glycans and glycoconjugates to those functions. It'll be a way for people to access this knowledge in new ways. So it'd be great, but it gets a bit complicated. There's that really bad word that I don't like to use in relation to glycoscience, but it gets complicated because the glycan itself might have a function that's completely independent of what it might be attached to, or it could change the function of the thing that it's attached to, or it may only have a function when it's on a particular protein. So figuring out how to encode all those sorts of details is taking more sort of deep thought and strategic planning than we thought it might have. But we've been pursuing this the past couple of years as a community effort through workshops really around the world, biocuration workshops and glycobiology workshops, engaging with domain experts. And we've been talking with Go Consortium as well, figuring out how we can help each other to encode these sort of more complex types of functions, and hoping we can build something that's really broadly accepted and ultimately impactful. I think in the next year or two, we should be able to roll something out. And this is just, I think, so important because it really going to help us connect to all kinds of other sorts of big data that we don't have a way to connect to right now, but connecting to through function is going to be, I think, really exciting.
I mean, to be fair, Michael, I feel like the whole reason why I ended up picking biology is because I did like that it was messy and complicated. So I hope that for the most part we don't shy away from something because it's complicated.
No, yes, of course. That's the beauty.
Kind of talking about sort of future looking and what's next for GlyGen. I'm sort of also interested to hear a little more about where you see things like AI and ML, helping in a safe way inside these, knowledge base workflows, and where it might be a little more risky to be using things like AI.
Yeah. So we are AI and machine learning, can be extremely helpful, particularly for tasks such as text mining, literature mining for publication. So that's an area we are actively using or developing AI approaches to identify glycosylation data, say, described in publications. Um, there are also other promising efforts, not just by GlyGen, right? There are other efforts ongoing worldwide on using AI and machine learning, for example, to help interpret experimental data, for example, automatically assigning glycan structures from spectra. So some groups are doing exciting work in that space. There are a few publications that have started coming out. So we are following those publications to see, how their data potentially could also be used in GlyGen. The known risk, of course, is hallucination. So that's at least where I see that one of the risks are. So AI system can generate highly plausible but incorrect information. We have all seen that while we have used maybe most probably ChatGPT, right? And other AI tools. So that's why I feel that AI outputs, they need to be grounded in structured knowledge and evidence, which connects to our previous provenance information and so on. So one approach we are pursuing is thinking about integrating AI with knowledge graphs. So in knowledge graphs, the relationships and evidence are explicitly represented through nodes and edges. And that structure can help constrain the system and reduce the risk for unsupported conclusions. So combining knowledge-based knowledge graphs with AI approaches and tools and models, I think can help us do things responsibly.
That's really a neat idea. So with the knowledge graph that you're kind of feeding into the AI, would that be kind of like a RAG then, where you're basically giving it like, here are the things that we already know and we want you to retrieve information from the knowledge graph that we're giving you in this case?
So yes. But it is, so there are two ways to look at it. So I'm going to give an example from the gene protein side. So everybody knows what BLAST, or everybody in biology has used BLAST, which is a sequence homology algorithm in use. So there is a default cutoff, right? So most of us, 99 or even more percent, 99.9%, will just look at anything which is above the cutoff. But a lot of discoveries actually have happened over the years below, underneath that cutoff, where people have found homologous sequences through additional methods. So one way to look at it is that, yes, some users will use AI to do things with and without hallucination, but then use the knowledge graph to confirm certain things as they move forward. The other way, as you are saying, is that constrain it right at the beginning and use knowledge graph right as a RAG method that you're mentioning, and use it that way. It again use what is it is being used for. I think making everything transparent is the key. And when the AI is presenting information, clearly showing where is that information coming from is going to be really important.
Got it. That's really neat. I hadn't thought about like knowledge graphs in that specific context. So I like that. So I would love to sort of have, especially for our listeners, if there's a lab that wants to contribute data, what is the simplest way to do that if they want to contribute to GlyGen?
Yes, so that's a very important aspect. As we described earlier, we are going out to all these databases and collecting information, and we have been very successful with that, which means, we have all the data, at least the one that is available in databases. But there's a lot of data published, as you just said, by people, and we want the data as well. So that's why we have worked on a submission system. So you can't submit the data directly to GlyGen because, there's all this data integration, harmonization, standardization, validation going on. But we developed a submission system called GlyTableMaker that people can use to deposit their data there. And then GlyGen, but also other databases, then can retrieve the information and integrate it in their own resources. So if people are interested in sharing their data, they can use that GlyTableMaker tool or get in contact with us. And we are very happy to help with getting their data out.
Awesome. Thanks for that. I wanted to, like, sort of my final question. I wanted to kind of think about, if I am a first-time user of GlyGen, right? This is the first time I've ever seen any of your tools, your resources, your website. Where would be a really good place to kind of start first, especially because you have so many amazing resources. Where's a good place to start as a beginner?
A good place to start would be the AI-assisted search. So users can simply type a research question in natural language, in English, natural language, and something like asking the glycosylation sites on protein X and see what the system returns. So it is an AI-assisted search. So in the back end, what is happening is that question is being translated into an advanced search query. So the user gets results. But as we all know, when we type in a question, sometimes we get results. We see that that's not exactly what I meant. So on top of the page, you will see modify the query. And there the user is taken to this advanced search page, and they can actually change what exactly the query is. Most people don't use advanced search based on what we have seen our logs, and in other resources also we have talked about. But what we do believe that using this AI-assisted query, people are doing advanced search in the background. And then if they get the results, that's great. They're happy and they move on. If they don't get the results that they're looking for, or the query was not interpreted correctly, we provide this option to modify the advanced search. And I think that was a great place for new users to start using GlyGen.
That does sound like a really great place, and I think I'm going to have to play with that some more. So, before we wrap up, I was hoping to ask more of a fun question for our very last question. And I'm curious to know if any of you have a favorite glycan.
Oh, I do.
Oh, let's hear it.
It's a core difucosylated trimanosyl chitobios.
Oh man, why is it your favorite, Michael?
Well, because it's only made in insects in the nervous system, and that extra fucose, I said difucosylated. Humans make fucosylated glycans a lot, but insects and some plants put another fucose on the core. And it's really cool because it causes allergies in humans, but it's really important to how the nervous system develops in flies.
Okay, that is a really cool answer. Renee or Raja, do either of you have a favorite glycan?
I don't have a
Totally fine if you don't.
I don't have a favorite glycan per se, but I have a favorite site. So,
okay.
So interferon gamma receptor 2 has this germline mutation in some individuals, which results in susceptibility to infection. And why is it my favorite? Is because that's how I actually started getting interested in glycoscience, because it's a genetic it's a SNP, single nucleotide polymorphism, right? So that's how I got interested. How that particular mutation leads to a gain of glycosylation, and that gain of glycosylation alters the way the protein behaves when it's trying to form a complex. And all of that has been solved in the past 15 years or so. But when I started, it was just a prediction, or it was found that mutation is there, and it was a gain of glycosylation site. But more recently, a paper came out in Nature which shows exactly what's going on. So for me, I find that site really interesting.
And what about you, Renee?
Yes, so I don't have a favorite glycan. I love them all the same, even the more complex ones. So
That sounds like a true answer from a loving parent. I love them equally. Well, thank you all so much for joining us today. I really loved hearing all about GlyGen, and I hope our listeners check out all of the amazing resources that you all talked about today. Thank you so much for your time, and have a wonderful rest of your day.
Thank you very much.
Thank you for having us.
Thank you. Bye, everybody.
You have been listening to Decoding the Data Ecosystem, a CFDE Training Center podcast. Be sure to check out our other episodes in this series. Find out more information about CFDE data and tools that may be valuable to your research by visiting our website.